Hello, this is Tessa Kegel, and this is HBS 5 OHC, Identifying and Controlling Hazards, Mitigation of Health Impacts, Neurotoxicity and Neuropsychological Issues. And this is Module 8, Part 2. This is the Commonwealth of Australia Copyright Act, which covers these lectures. Mechanisms of action. Unfortunately, the pathophysiology of most conditions related to neurotoxic exposures is not very well understood. Much of the current knowledge has been extrapolated from events such as industrial accidents, which have provided an opportunity to observe the effects of exposures on humans. As can be imagined, it's very difficult to implement human trials under controlled conditions. Neurotoxicity mechanisms. Neurotoxins have a range of different effects. Chemicals such as chlorine have the effect of impairing oxygen supply to the cells. Carbon monoxide binds to hemoglobin and other iron compounds, thereby reducing the oxygen carrying capacity of the body, as well as binding to cytochrome oxidase and thus directly inhibiting mitochondrial respiration. Organophosphates inhibit acetylcholinesterase, causing accumulation of the neurotransmitter acetylcholine at the cholinergic synapses, while methyl and butyl ketone reduces ATP in nerve tissue, thereby disrupting axonal transport and repolarization of nerve fibers and therefore nerve conduction. Temporal lobe brain structures have been found to be particularly sensitive to exposure to trichloroethylene, with even brief exposures resulting in cognitive impairments, as well as selective neurotoxicity on the trigeminal nerve. Carbon disulfide interferes with enzymes containing sulfonyl groups. The possible effects of solvent exposures have been defined as structural, biochemical, and functional. Deficits and symptoms will vary depending on whether spinal cord, peripheral, or central neurological structures have been affected. Junterman also suggests that this may be different in developing, degenerating, or aging nervous tissue. This table provides a little bit of detail about neurologic symptoms and signs. So first of all, the syndrome, then the neuroanatomy, symptoms and signs, and some examples. Now, I'm not going to go over all of these. You can read these yourself. However, I'll just go over one of them. Motor neuron disease, spinal cord neurons in terms of the neuroanatomy. Symptoms and signs are muscle atrophy and weakness, and some examples of exposure are lead and manganese. And I'll let you review this table at your leisure. Another factor important for the OHS practitioner to appreciate is that symptoms can be variable in degree as well as fluctuate over time. Unlike the symptoms associated with most other health conditions, neurological disorders can be associated with over 100 different types of symptoms. Additionally, individuals vary in chemical uptake due to factors such as ventilation rate, skin perfusion, genetics, effects of alcohol, which, for example, lowers the threshold for toxic effects of lead exposure. Synergism between chemicals is also an important consideration due to the potential for multiple exposures at a work site, for example, working with several different types of solvents. For some toxins, sufficient evidence has been accumulated across a number of different studies to identify the difference between acute and chronic exposures and impacts on the neural system. This table considers the health effects of both acute and chronic exposure to various neurotoxins. So it goes through a range of different toxins, so carbamates, ethylene oxide, manganese, organotin, thallium, toluene, and trichloroethylene. And it considers both the acute exposures and the chronic exposures. And I'll let you read that table at your leisure. Most of the international documentation of exposures have been based on recording the immediate effects of acute exposures, usually following a major incident, rather than the identifying effects associated with gradual occupational exposures. For this reason, there is less evidence of the more subtle effects of chronic dose exposures or of the long-term effects of acute exposures. This is recognised by the WHO and the Nordic Council, who established a protocol for diagnosing the effects of exposure to solvents. They specified differing test batteries for acute, mild, chronic, and severe chronic encephalopathy. Repeated acute exposure to solvents have been shown to result in the same effects as chronic dose exposures, which can be permanent. Generally, it's common for exposures to result in psychomotor effects, such as muscle weakness, tremors, sensory loss, and uncoordination, predominantly through effect on the peripheral nervous system. Sometimes there's recovery following cessation of exposure, as there's reduction in the body burden of the chemical. Commonly, these also affect a person's cognitive and perceptual functioning, including memory, attention, and concentration. A number of substances have had some neuropsychological evaluations following exposures of some sort, including lead, manganese, cadmium, organotin, mercury, pesticides, carbon monoxide, chlorine, ethylene oxide, formaldehyde, methyl bromide, hydrazine, hydrogen sulfide, organosulfates, trichloroethylene, tetrachloroethylene, toluene, diisocyanate, carbon disulfide, and styrene. And once again, I do apologise for mispronunciations. In contrast, deficits in cerebral functioning related to verbal and language domain areas are less common. We cannot confidently draw conclusions as to the effects on executive function, as the extent and quality of testing in this area is very limited. There is some evidence of effects of chemicals on brain functioning relating to visual processing. Processing of visual material encompasses a range of capacities, such as visual perception, visual tracking, visual motor coordination, and visual memory. Unfortunately, the tests used in research have not been particularly thorough in specifically determining the nature of deficits in this area, often using a single test, such as replicating a pattern using blocks. With more thorough testing in this area, some quite distinct deficits may be found to be associated with particular chemicals, such as tri- and tetrachloroethylene, chlorine, and some of the heavy metals. Potentially, the most challenging exposure assessment area is determining the effects of neurotoxins on a worker's memory and learning skills. In this domain, it's possible to determine distinct effects between chemicals. For example, between heavy metals with effects on attention and immediate memory for lead, spatiotemporal sequencing deficits for cadmium, verbal memory for organotin, and visual memory for mercury. Pesticides showed deficits for visual memory and immediate recall of information. Organosolvents showed slower learning, especially for linguistic information and decay from short-term memory. Styrene indicated deficits in short-term memory and memory for verbal information. Carbon disulfide indicated memory deficits for poorly structured information, while trichloroethylene exposure indicated deficits in learning new information and in recall.